Well, if you don't know about chestnut, chestnut is a big tree, and it used to call the king of the forest. And that chestnut is gone almost completely by an invasive fungus accidentally imported from Japan after, not after, is somewhere late 1800, and for the beautification. When it comes to New York Zoo, and then the first, the bacterial blood disease reported in 1904. And if you see, the chestnut growing area is about 200 million acres, 4 billion trees, up from Maine, south, up to, say, northern Mississippi and Louisiana. And that's entire the tree gone in a span of about 50 years' time. You can see that's how the invasive species can destroy our cultivated species. So it's not a foundation of working to transfer the resistant gene from Chinese chestnut to American chestnut. American chestnut is huge, tall, less branch tree compared to the Chinese chestnut. And so that was the main goal, to back it in forest. When you cross with two different species, there may be always some chromosomal irregularities due to inversion translocation. And then they started working in early 80s, and then they found out now they have a problem. They never checked the chromosomal morphology or chromosomal behavior. Then what happened with this genetic map, they found two of the linkage group giving a distorted map. So there was a speculation there could be a reciprocal translocation either of these two species. Then they just asked our group to work on and see what we have in chestnut background. That's the brief background about chestnut work. So first, that introduced in 1876, and this is the Chinese chestnut or Japanese chestnut. That's in Connecticut area that's first grown, and the blight disease reported in 1904, and by 1929, the report said it's covered all over the chestnut growing area, and then by 1950 it's gone. And we know plant quarantine law. That law passed based on this man-made disaster to prevent that's happen again with any other crop. And the Canadian Wildlife group that designated that it's a threatened species. Well, that's the statement. That's a man-made disaster. It's happened, but man didn't do it intentionally. It happened accidentally. Chestnut tree, doing the breeding is not, say, rice breeding or wheat breeding, and its life cycle 5 to 6 years. But in rice or wheat, we can harvest 3 crops in a year. Now you see how difficult that breeding is. And I said that. No. I was looking for one slide, that is gone. Then we take the flower bud, we dissect that, and then analyze the myocyte. And what it showed, the Packaging analysis, it showed, yes, there is a reciprocal translocation. This is a Packaging spread, and the configuration it showed, and it fits with this reciprocal hypothesis. And this is another cell, and if you see that, it's clearly showed that it's a reciprocal translocation. Reciprocal translocation is just chromosomal segment exchange between two non-homologous chromosomes. But the plant produce normal activities. Normal seed you cannot notice with the normal plant and reciprocal translocated plant. Nothing is losing there. But when you cross between one reciprocal translocated plant with the normal plant, there's the problem comes. And here you can get the pairing, and it gives about 33% viable gamete and about 66% non-viable gamete. Now, depending on which one is transmitted. Just showing it, we get 1/3 balanced and 2/3 unbalanced gamete. I'm not going to go there in details, but if is a 2 gene controlling this chestnut blight, and if happen to be those 2 genes, they're on the reciprocal translocated chromosome, then it's mess up the breeding program. So we need to find out where are those 2 guys sitting. And that's the work we're doing, and we are close to find out who is 2 non-homologous chromosome exchange that on. And this metaphase, it shows it's a quadrivalent, and it proves that, yes, it's reciprocal translocation there. In the background, there is also trivalent. There's 3 chromosome involved, and this configuration, it shows it's a 3 chromosome involvement. It's not only one reciprocal translocation. There is another mess-up event happen in chestnut. Then that work led us to do some more work, the structural organization of that genome. What we did, this is a F1 between Chinese and American cross, and the nuclear organizer chromosome or satellited chromosome, it shows Chinese chromosome is different than the American chestnut chromosome. And if you see that this satellited portion here is a little bit larger than the American chestnut, and it shows here. This is a genomic in situ hybridization, that what I described earlier this morning. And here it showed Chinese satellite is much larger than American American chestnut satellited chromosome. Satellited chromosome says that where is the nuclear organizer region is in sorghum, it's here, and that's the satellited region. And there's a diagrammatic representation just showing which one we call the satellited chromosome. Just to recap your undergrad coursework. Then we started number of Chinese chestnut, and what we found, they differ in satellited chromosome. And also this one is also new findings, that is colocalized the 5S rDNA with the 18S rDNA in satellited chromosome. The variation we found, if you see Basiliki, Donsoret, copper, and queen, and these 2, they look same, but the heterochromatin composition here, this area is little bit lighter than here. So these 2 line differs. That is completely different. And this is heterozygous for 5S. And if you see, this is homozygous for 5S, but satellited region is also differ in this line. Chestnut is a cross-pollinated crop. Then we did that. After that, we are working with chromosome by chromosome to do the integration for that genetic map into chromosome. This is an example of sorghum we did, and we using the same technology to do the chestnut structural organization. Chestnut is a hard crop compared to cereal crops. It's about, I should say, somewhere about 25 to 30 times harder than any cereal you can work with. It takes a lot of effort to do the cytology in chestnut. This is a, we took, say, about 2 to 4 marker to match the linkage group with individual chestnut group, chestnut chromosome. Then doing all this, we assigned all the 24 chromosome, 12 chromosome of chestnut by doing this way for back hybridization. This is LG bee backfish. We use 4 probe, and then interestingly, it shows, if you see that one marker is very close to the centromere, and that shows that it could be highly linked with the disease-resistant gene. If that one is close to the centromere, to transfer that disease-resistant gene from Chinese to American chestnut is going to be harder. But then it comes to all these people that's doing the transformation. But doing transformation is chestnut is a nightmare. But I think that would be possible doing a lot of work. So this one I showed before. Okay. The satellited one, this is sorghum chromosome. About 25% of the satellited region and the null region was not mapped earlier, but with this cytology, we mapped that region in chestnut. So it's started from here, the genetic map, and up to there, but that mass were not mapped due to highly heterochromatic. And this slide clearly explain, demonstrate that how we map this portion of the chromosome. This is a first fish. Then we thought it would be the nucleus organizing region. Then we used the 18S, and then it showed, yes, it's the linkage group A contained that 18S site of that chromosome. And that's what we said about this mass were not mapped in genetic analysis. And we use about here, I do not know, 15 or 14 back probes to go with that whole chromosome. And this is interesting. In trees, for chestnut, is distributed along the chromosome, not like sorghum or rice. It's across the chromosome. There's all this marker is landing on the chromosome. This is LGA chromosome, and this one is linkage group B chromosome. And some are saying it's close to the precentimeric region. If the marker is here, then it would be hard to transfer from this area. And that shows the complete characterization. Oh, well, I shouldn't say complete characterization. It gives us a sense how they are distributed in chestnut tree. So I'm just showing this this image because you need the toys, the microscope. If you do not have the right instrument, it's going to be harder. And our lab is highly resourceful about all the instrumentation you need, doing all this cytological work. So in summary, the reciprocal hypothesis is confirmed. Chinese chestnut chromosome contain that major 18S compared to the satellited chromosome compared to the American chestnut chromosome. And it has structurally different that American chestnut chromosome. Identified second locus of 5S RDNA, and 4 variants of satellited chromosome identified in chestnut, Chinese chestnut. Satellited region contains highly heterochromatic DNA in Chinese chestnut. 12 linkage map assigned to a specific chromosome in chestnut. And that major 18S site has been assigned to LGH. In acknowledgement, the fund from Chestnut Foundation and our institute, then that is Port Service, and physical facilities coming from the Texas A&M. I'm located at Texas A&M campus. And people involved, this Chestnut Foundation, Paul, Cisco, Fred, Robert, William, and Laura. In our group, Dana Nelson, he is the project leader, Tom, Majid, and Harsha. Ron Clay from Minnesota, he is in this cytology section. Time to time, we get some advice from him. I think that's all. Thank you for your time. And if you have questions, I'll try my best.